Hi everyone, my name is Parvati KS and I am pursuing my MSc Biotechnology from Life Science Department, Christianity College, Bangalore. This video depicts the activity which is assigned by our faculty, Dr. Manikandan, Faculty of Department of Life Science, Christianity College, Bangalore. In this video, we are going to discuss about DNA microarray principles, procedure and applications. Coming to the introduction part, so what is initially the DNA microarray? So these are the solid supports which are made up of glass or silicon material in which the DNA we are going to attach in a grid fashion manner. In this DNA spots, each represents the single gene and it is known as the probe. So in each array there is a single gene and it is all now known as probe. So these microarrays help in the analysis of 10,000s of genes simultaneously in a single process, and these are also known as DNA chips, gene chips, DNA array, gene array or biochips. So coming to the microchip. So each gene or a probe which is occupies on the spot and the chip which varies a level of gene activity which is introduced in the genetic material. So these microarray use the technique called hybridization and dictate a specific DNA or RNA in the sample. Here we are using fluorescently labeled target sequence that will bind to the probe and later generates signal. So coming to the principle, on what basis this DNA microarray works. So the main principle of this DNA microarray lies on the hybridization technique between the nucleic acid strands. That means the property of complementarity of the nucleic acid. That means the pair with each other, the DNA or the nucleic acid pair with each other by forming hydrogen bonds between them according to the complementary nucleotide base pairs. For this, the samples will be labeled with fluorescent dyes and at least 2 samples are hybridized to chip. So the complementarity of the nucleic acid sequence between the sample and the probe attached on the chip get paired via the hydrogen bond. With the help of this hydrogen bond, the complementarity takes place. And the non-specific bonding sequence, that is while washing unattached DNA strands will be there. After washout, this during the washing steps can be removed. Next one is the fluorescently labeled target sequence that bind to the probe sequence generate a signal, and these signals mainly depends on the hybridization conditions such as temperature. And after washing this, the hybridization product, while the total strength of the signal which depends on the amount of target sample present. Using this technology, the presence of one genome or the cDNA sequence in 1 lakh or more sequence can be screened in a single hybridization. So this is the normal picture which depicts the DNA microarray process. So here we are taking 2 samples, the cancerous sample and the normal samples. From this, the mRNA is extracted. From the mRNA, cDNA is produced via reverse transcription, and later it is labeled. And these are hybridized DNA probes with oligonucleotides on the microarray. And this depicts, these colored one depicts the results that we get after the fluorescent signals that we are collected. So here the gray one represents no cells in the chip, and the yellow represent both the cells, and the green represents the normal cells only, red represents the diseased cell only. In this way we can come to an conclusion. So coming to the type of microarray, there are mainly 2 types of DNA microarray process, that is cDNA based microarray and oligonucleotide based microarray. What is cDNA based microarray? So this is also called spotted DNA arrays. So here the chips are prepared using cDNA. These cDNA chips or cDNA microarray is also called probe DNA, and the cDNA are amplified using PCR. So here we are using a PCR product for the DNA microarray process. These cDNA are then mobilized on a solid support which is made up of nylon filter or a glass, and the probe DNA are loaded into the cDNA in the solid support and get spotted. Now the small volume of this DNA prepared is spotted on the solid surface and by making the physical contact between them, and the DNA is delivered mechanically or in a robotic manner. So this is the pictorial representation of the process, the spotted microarray. That is the array preparation and the target preparation. In the array preparation, we are collecting the cDNA or the oligonucleotides and are assigned by robotic manner or mechanically. And then the target sequence, that is the sample A and sample B's are collected, and they are labeled with dyes, fluorescent dyes. Here we are using cyanin 3 and cyanin 5. Cyanin 3 is labeled on the normal samples and cyanin B is labeled on defected sample, and these samples are then hybridized in the chip and get imaged. The data is analyzed and then we will come to one conclusion. Coming to the oligonucleotide array. In oligonucleotide array, we are using short DNA oligonucleotides which are spotted on the array. That means the number varies from 20 to 25 mers per gene. So the main feature of this oligonucleotide microarray is the gene is normally represented by more than one probe, and these are enabled by photolithography from the computer industry off the shelf. So what are the requirements of this oligonucleotide array? So here we are needed the DNA chip, target sample which is fluorescently labeled, dyes, fluorescent dyes, probes and a scanner for analysis. So first of all, we want to collect the samples. Those samples can be collected from the tissue of the organism that we wish to study on, and we can collect two types of cells, healthy cells and defected cells, for the comparison of the results. Next one, to isolate the mRNA. So we want to isolate the mRNA from the sample. For that, the RNA is extracted from the sample using phenol or chloroform, and we are extracted and separated the rRNA, tRNA from the mRNA. So these mRNA is tailed in the column beads using to bind the mRNA, and after the extraction of the mRNA, the column is rinsed with buffer and the mRNA is isolated from the beads. So then to create the cDNA, we will make use of the reverse transcription method. From the mRNA, we will get the cDNA strands. So with the help of this, it can be easy to distinguish the sample category of the cDNA. Next process is nothing but the main principle behind this process, that is the hybridization. Here the cDNA from both the samples are placed in a microarray so that each cDNA get hybridized to its complementary strands, and also we get thoroughly washed to remove the unbound sequence. Now the last process is the collection and analysis. In the collection of data, we are done using a microarray scanner, and the scanner consists of a laser, computer and a camera. The laser will be excites the fluorescence of the cDNA, which will result in the generation of the signals. When the laser scans the array, the camera records the image which is produced from the array, and these are stored as data and provides the results immediately after the process, and these are then analyzed. So the difference in the intensity of the color of each spot determines the character of the gene in the particular spot. So these are the main difference between the oligonucleotide microarray and cDNA microarray. So in oligonucleotide microarray, we are only using a small oligonucleotide sequence, but in the case of cDNA microarray, we are using a PCR product for the results. So these are the basic principle and the process of the cDNA process of the DNA microarray, the principle and procedure. And coming to the application, mainly this can be determined to use how the particular disease affect the pattern of gene expression in various tissue, or to identify the infected organism. Thus in clinical medicine also we are using these techniques and this potential for diagnosis. Beside this, it is applied in many fields such as discovery of drugs. So it plays a very important role in the discovery of drugs, diagnostics and genetic engineering fields. It is also help in the alternative splicing detection, proteomics, functional genomics for the sequencing of DNA. Gene expression profiling is done in the help of DNA microarrays, and it also help in the toxicological research studies. So these are my references which I followed for this process complete. And thank you for giving me the chance of explaining this process in details. Thank you for the faculty, Dr. Manikandan, for this chats. Thank you all.